And for the first session, may I request Dr. Monica Peter, who will talk on experimental model of acute and chronic pancreatitis. Dr. Monica is from Assistant Professor, Christian Medical College, Department of Biochemistry, CMC Vellore.
Alcohol and diet-induced models. So for today, among the other variants, I'll be talking about the diet-induced model. So first, let's start with the cerulean model. So cerulean is a secretagogue analog. It acts by binding to the CCK1 receptors and it activates them. And like the talks that was given today, there is premature activation of the zymogens, and it is known that in cerulean model, acute pancreatitis, that is only a mild edematous pancreatitis that is usually observed. However, there was a recent study in 2018 which was published, and they have shown that when you increase the duration and the dose of cerulean, there is actually a severe form of acute pancreatitis that develops. So, if you are looking for a model that has which is commonly used, it's cerulean, which is commonly used, it's highly reproducible, and you could use it for studying various conditions. So the next model that I'm going to talk about is the basic amino acid model. So among the basic amino acid model, it's the L-arginine which is commonly used, and this is the dose which is commonly used for experiments in rats. For mouse, it's slightly different. And one advantage is this model resembles the human necrotizing pancreatitis. The disadvantage is there has been severe mortality observed in mice which were induced with L-arginine, and that is one thing. So we have to be very careful with the dose that we use for inducing the pancreatitis using L-arginine. The next model is the retrograde duct infusion model. So for this we infuse the bile acid through the pancreatic duct, and it has a non-homogeneous distribution which is usually seen in humans, so you can use it. So however, since it's an invasive procedure, there are technical challenges which are associated with this. And last model is the diet model. So it is the choline. The diet is actually deficient in choline and it is supplemented with ethionine. So this leads to development of acute pancreatitis, but it usually takes some 2 to 3 days for the acute pancreatitis to develop. It's one of the least invasive model among all those that I have discussed before. So in our lab for the past few months, we have been working on establishing a severe model of acute pancreatitis, and our objective was to have a model that replicates the systemic inflammatory response and the endothelial dysfunction. So based on the discussion today, endothelial dysfunction in fact plays a very crucial role in the disease progression. So we wanted to create a severe model of acute pancreatitis. So with that objective, we proceeded with the two models that we have discussed so far. So we looked at the cerulean and the L-arginine model. This is the protocol that we used in our lab. So if you look at, so we use the C57 6J 10 to 12 week old male mice, and let's look at the first column, which is the cerulean model. So if you see the cerulean model, the mice were under fasting conditions with ad libitum access to water. So on the day 1 of the experiment, the mice were injected with intraperitoneal injections at a given dose for 8 consecutive hours, hourly injections. So we started at 0 hour, and for 8 continuous hours, every hour the mice were injected with the given dose of cerulean. Similarly, we had a control mice which also received saline. It was also treated similarly, but it was given saline. And the mice were allowed to be kept for observation with access to water. However, they were kept under a fasting condition. On day 2 of the experiment, we actually gave them again 8 consecutive hours of injection, and 1 hour after the last cerulean injection, the mice were sacrificed and tissues and blood samples were collected. So looking at the arginine model, it's relatively simpler to perform from our experience. So what we should do is, on the day 1 of the experiment, we give injection at 0 hour, and after 1 hour of the first injection, we give the second dose, and 72 hours the mice were kept for observation, and after 72 hours, the mice were sacrificed. So unlike the cerulean model, in the arginine model, the mice were allowed to have free access to feed and water. So let's look at some of the enzyme activity that we measured in the lab. So if you look at the Serlin model, so I would like to mention that there is no simple reference range or a cutoff that can be used like we have in humans to look at the enzyme activity in animals. So it's always better to have a control group so that you can say whether there is increased enzyme activity or not. So similarly in our study also we have actually tried to have a control group where the serum amylase and serum lipase levels were extremely high when you compared with the control. And the arginine model also showed a high increase in the serum amylase and the serum lipase levels when you compare with the control. So this is just a very starting thing that we have begun in our lab. So just one animal in each group were used. So we are still working on increasing the sample number. So let's look at the histopath. So if you look at the pancreas of the Serlin model, so we actually send the histopath report to the pathologist, and based on his report, so what he suggested was there is definitely mild to moderate interstitial edema in the pancreas of the Serlin model, while there is no specific lesion in the control mouse. So if you look at the arginine, definitely there was even moderate to, you can look at here, so moderate to marked intraacinar edema that was appreciated in the L-arginine model, increasing severity of pancreatitis in this particular model. So when we looked at the pancreatic tissue, it was relatively normal with no lesions. We also wanted to look at the other organ like liver. So if you look at the liver, we did not find any significant difference between the two, that is the Serlin or the control, apart from some droplet steatosis which was observed in both the Serlin as well as in the control models. So even in the arginine, we did not find any typical finding that we were expecting to look in these models. So there were no systemic changes that we had expected that we would see in both arginine and also in the Serlin induced models. So let's quickly look at the experimental models of chronic pancreatitis. So among the frequently used chronic pancreatitis, so based on this paper, repeated Serlin induced model, we have the alcohol with lipopolysaccharide, duct obstructive model, duct infusion, we have the genetic models and also the immunologic models that can be used. So I'll be going through the advantage and disadvantage of this model one by one. So if you look at the repeated Serlin model, it actually mimics the recurrent episodes of acute pancreatitis that could eventually lead to progression to chronic pancreatitis. It's a highly reproducible model. The second one is the alcohol with LPS. So if your objective is to look at the alcoholic type of chronic pancreatitis, this is actually a good model to use. So that it mimics the human alcoholic induced chronic pancreatitis. However, some studies have reported that this leads to systemic toxicity. The next one is the duct obstructive model. So where the duct obstructive and the duct infusion, so here you can actually use them to study the biliary type of chronic pancreatitis, so gallstone induced chronic pancreatitis, and both are invasive procedure and hence there are definitely technical challenges associated with them, and in some cases the mortality could be higher. Genetic models, we don't have much about them, so I'm sure there are other talks where they're going to talk about these models. So these models can be used especially in hereditary pancreatitis. So the disadvantage of using such model is they are expensive, time consuming, and usually the human pancreatitis is a polygenic. However, when you establish a model, it will be a single gene focus, so that could be one drawback. Last is the immunologic model. If you are looking at the autoimmune initiated pancreatitis, this model can be used. But however, I think it's still done under different labs and it's not fully thoroughly characterized. So to conclude, there is not a single experimental model that has been developed that can fully replicate the human pancreatitis. There are simpler models which could be easier to reproduce, however they are physiologically not quite relevant. However, there are complex models which are time consuming and resource intensive. So to conclude, the choice of model for inducing acute pancreatitis should be based upon the research question, your study objective, and what clinical scenario or pathological mechanism is under investigation. So all these factors must be considered, and you need a tailored approach before choosing an appropriate model for inducing either acute or chronic pancreatitis. So that's the end of my talk, and thank you everyone for your valuable time.
Monica, I have a question.
Yes.
When you are developing serine and arginine models, you are using the same type of marks, right? In serum lipase, the control values are 9. In the arginine model, it is 5 times more. Is there any explanation for that? I know it's possible.
Yeah, we don't have any explanation for that. Even we were like, after looking at the results, we were wondering if there could be any reason why, specifically in the L-arginine, even in the control group, we have such marked elevation. So actually, we are not very sure why it has happened. So it's our first experiment that we have done, and so we could not come up with the possible conclusion of why.
Even the cases are also, it's double.
It's double, yeah. So amylase is comparable, but the lipase is quite high in the arginine model. So both in the arginine and also in the control, when you compare with the serine, the values are very...
control is saline,
yeah.
so there is no intervention. It's similar.
It's
in
similar.
serine model as well as in arginine model, it's the same.
Yeah.
Actually, the level of lipase does not correlate with the severity of the disease. So whatever the level is there, you're fair enough. You have to just establish that the pancreatitis is induced. It does not correlate with whether it is a 15,000 or 9,000. It does not make any difference. Any question?
Monica, have you thought of trying in vivo imaging of the organs, like using IVS imaging system rather than end of the experiment?
Yeah, so I think first our objective here is to establish a severe model, and then probably when we are actually looking at the endotheliopathy, like, which is very typically seen in pancreatitis, like we want to look at the blood flow and the coagulation, some imbalance. So all that I think we'll need the IVIS. So first we would like to establish the severe model, and then we will go for all of that. Yes, of course we have plans to do that in the future.
Thank you.
For chronic also there is L-arginine model, which is very slow incremental dosage, continuous. That's what we are publishing our paper. Dr. Saluja's group has validated the chronic pancreatitis induction protocol.
Sure.
Any other questions? Yes.
So how does it activate? So we don't know the full complete mechanism of how it induces pancreatitis. So what we know is L-arginine, it is usually the precursor for nitric oxide. So when you give that in excess, it can actually lead to production of ROS, so the reactive oxygen species. That in turn could trigger, like for example today they were showing NF-kappa B activation. So it could lead to inflammatory cascade activation that in turn could cause the events of acute pancreatitis. So whether it's completely understood, I'm not very sure, but this is what we know from the from the papers that have been. Cerulin. So cerulin is actually a secretagogue analog. It binds to the cholecystokinin 1 receptor and it activates the zymogen. So basically premature activation of zymogen, trypsinogen.